This is Sophie Heller, and this is an ACT science question. The question is asking us which experimental procedure was used directly, so this is the keyword, directly, to determine the pH of a blood sample. So what the question is going to trick us up with is it's going to mention things that are used in these experiments, but we want to see what has the direct influence on measuring this pH in the blood. So if we look at experiment 2, we see that the pH of blood is determined using another blood subsample and a glass microelectrode. This is how the pH of blood was determined. So this is what ties into that idea of directness. So we know that we need the microelectrode to directly measure the pH of blood. So let's look at our choices and see what we can make of that. Choice 1, no experimental procedure was used, the pH must be calculated. So this is directly contradicting the idea in experiment 2 that it was calculated using a subsample and the microelectrodes. That's not valid. Choice 2, the number of blood cells present was counted. And we see that present in experiment 3. In experiment 3 is evaluating one of the pHs determined and reasons for why it seemed higher than normal. But this isn't a direct result in order to find the pH. This is just something about a specific, really elevated pH. Choice number 3, we see the glass microelectrode. That's going to be our correct answer. And choice number 4, a microgasometer was used, and that's another thing that we see in experiment 2, but it's used not to find out the pH of blood, but to collect the carbon dioxide. So that's not good either. So choice 3 is our answer.